Welcome to the BLAST tutorial series brought to you by the Genomic Education team at the Jackson Laboratory. This tutorial is a demonstration on how to compare or align two or more DNA sequences using Nucleotide BLAST. BLAST is a powerful tool that allows you to compare and identify variation between two or more DNA sequences. BLAST will use sequence alignment to accomplish this. Before beginning, it's important to have the DNA sequences you want to compare handy. I have mine here in a text file. To then start the comparison of the sequences, navigate to NCBI's BLAST using your browser. From the BLAST main landing page, select Nucleotide BLAST here on the left under Web BLAST. Once Nucleotide BLAST loads, check the box Align two or more sequences to load a second entry box, which will allow you to compare sequences. Without checking this box, you will not be able to compare two sequences. In this tutorial, we will compare a few different gene sequences with a reference sequence for that gene in order to determine if there is variation in any of our subject DNA sequences. A reference sequence is considered a standard sequence, or in this case, a sequence without variation. To do this, first we must enter the reference sequence into the large box at the top, Enter Query Sequence. This will be the query sequence. So I'm going to copy and paste my reference sequence and paste it here in this Enter Query box. Remember to include the description line, which begins with the greater than sign. Then copy and paste the sequences to compare in the box below labeled Enter Subject Sequence. You can enter one sequence into this box or multiple sequences. If you are entering multiple sequences, you can put them in one after another, each starting with the description line. It's important to include the description line because this will help you distinguish between the two sequences once they've been aligned. I'm copying and pasting 3 sequences into the subject box now. You can see there are 3 sequences, each with a description line. You can leave all of the settings the same and then go and hit the BLAST button. The page will refresh with results including the sequence alignments. Once this happens, scroll down to the table and stay on the Descriptions tab to see various aspects of the alignments to the reference sequence. Now I did 3 different alignments, which you can see here, but if you see one, you should see one alignment for every sequence that you entered into the subject box. Let's look first at the percent coverage, which is indicated here by query cover. In this case, all 3 alignments have 100% coverage. Coverage is an indication of the length of the compared query and the subject sequences relative to each other. 100% coverage indicates that the subject sequence spans the entire length of the query, which in this case is the reference sequence. Next, let's look at percentage identity, which is abbreviated here, per ident, which provides an indication of how similar the subject sequences are to the query sequence. More specifically, this value gives you a clue as to how many of the nucleotides are the same between the two sequences. 100% indicates an exact match. We can see here that one of the sequences matches the reference 100%, but the other 2 are less than 100, meaning there must be differences between those sequences and the reference. Now let's click the Alignments tab to see how many of the DNA bases in the sequence, or how the DNA bases in the sequences compare. Change the view of the alignment to pairwise with dots for identities. This will provide a view that is easier to visualize the differences between the two sequences. And scroll through the alignments to locate differences between the subject and the reference sequence. Let's look at the first alignment. Remember that the query sequence, which is here in the top line, is the reference sequence, and the subject is one of the sequences we entered into the subject box. Look at the top of the alignment to see which one. The description line should be listed here. The alignment starts at DNA base 1, and there are numbers at the beginning and end of each line to indicate the base number. The bases that are the same between the two sequences are depicted as dots on the subject sequence. So it doesn't look like in this first alignment that there are any differences, as there are dots for each one of the nucleotides in the reference sequence. Scrolling down to the second alignment, we can see a red T in the subject. This means that a T is a difference between the C that is in that position on the reference sequence. In the third alignment, we see two places where there are differences. We see the same C to T substitution that we saw in the sequence above. We also see this place where there are dashes in the reference sequence and red T's in the subject sequence. This indicates that there was an insertion in the subject sequence. The dashes are placeholders for bases that don't exist in the sequence. If the dashes had been on the subject line, then that would indicate a deletion in the subject, and the bases would be present in the reference sequence, but not in the subject sequence. Thank you for listening. For more information about or for other tips on using BLAST, please see the other tutorials in this series. Also, check out more resources provided by Jack's Genomic Education on our website.